agree with the speaker, the webinar will be recorded, and I started now recording the webinars. And so everybody should agree, should accept this. The recorded version of the webinar then will be uploaded on the iPlanta webpage, and it will be available for everybody, open access. And then we will have the also the speaker accepted to send and share the PDF of the presentation, so that it will be also available on the iPlanta webpage. During the webinar, I think that I suggest everybody to keep clearly the microphone off and possibly also the camera off. This will make more easy the system to run. As usually, the chair of the webinar, Olivier, will collect questions that can be posted on the chat and transfer the question to the speaker at the other hand of each presentation. And then at the hand, we will have a general discussion that will be always managed by Olivier. As usually, I will ask at the hand to please remember me in case I forgot to turn on the camera, so we can also have a group picture, virtual group picture that is always well appreciated by everybody. And so just to show your presence at this webinar. Well, thanks again to Olivier and to all speakers for this availability, and I leave you, Olivier, the floor to start, introduce the speaker and manage the webinar on pest diseases controls through RNAi technology. Many thanks.
So good afternoon, everyone, or good morning or good evening, depending on where you are. So as Bruno said, welcome to the iPlanta webinar. We have an interesting schedule today, consisting of a number of presentations from experts on insect RNAi, who will share some of their work related to RNAi efficacy. You'll see that most presentations have to do in this context. As we'll see shortly in the first presentation, RNAi efficacy in insects is quite variable. And there are a number of different barriers, both physiological and cellular, that could negatively impact sensitivity of an insect to RNAi. And this is what most presentations will delve into today. So in this webinar, we have a series of presentations looking at this variability, the different barriers, factors that are involved. But we also have presentations that look at delivery of Dsrna to overcome some of these barriers. And finally, we'll also discuss how this variability in different populations or in different species, these barriers, how they could also potentially lead to the emergence of resistance against exogenous delivery of Dsrna. So today we have 7 presentations, all around 15 to 20 minutes long. So as Bruno said, if you have any questions, feel free to write them down in the chat. I will collect them and then pose the questions. This could be either after the presentations themselves, depending on how much time we have left, or I will keep them, save them till the end of the webinar, where we have a 20-30 minute discussion time available for us, and I can pose the questions then. So our first speaker today is Dr. Anastasia Cooper from Kansas State University, and she will give a presentation on RNAi efficacy in insects, different molecular systems or different barriers affecting RNAi efficacy. So the floor is yours, Anastasia.
Thank you for that introduction. Give me a second to get things set up here. All right. Well, thank you for the introduction. And today I'm going to be talking about mechanisms that are limiting the efficiency of RNA interference in insects. So for my talk today, I'm going to give you a brief background on the history of RNAi and its uses in insects. Then I'm going to talk in detail about the 5 mechanisms that are limiting RNAi efficiency in insects. And then I will close with a brief conclusion and perspective. So RNA interference, or RNAi gene silencing, was first described in animals in 1998. And then a few years later, the lead authors received a Nobel Prize for their discovery that RNAi can be exploited by experimentally introducing double-stranded RNA to silence specific genes. In 2007, there were 2 papers that showed proof of concept for the use of RNAi in pest management. And then about 10 years later, the EPA approved the first transgenic crop for control of insect pests using double-stranded RNA. And now there's tremendous interest in using RNAi as a new mode of action to control insect pests. Unfortunately, not all insects respond equally well to double-stranded RNA. There's some organisms like the western corn rootworm or the Colorado potato beetle that exhibit very high RNAi efficiency regardless of double-stranded RNA delivery method. Then there are other insects like the migratory locust or the German cockroach that really only exhibit high RNAi efficiency with injection, but not with feeding. So with these species, after injection, you get very high levels of gene suppression, very clear phenotypes, and even mortality depending on what genes are targeted. However, there's a third group of insects, such as the spotted winged Drosophila or the European corn borer, that are highly refractory to RNAi, regardless of double-stranded RNA delivery method. And it's because of this variability in RNAi efficiency among insects that mechanistic research has been needed to elucidate the factors influencing RNAi. So according to the literature, there are 5 mechanisms that are likely influencing RNAi efficiency in insects. So the first is instability of double-stranded RNA in the hemolymph, in the gut contents, and sometimes the saliva due to nuclease activity. And the second mechanism is insufficient double-stranded RNA internalization, either through endocytosis or through the SID transmembrane channels. And then the third mechanism is possible deficiencies of core RNAi machinery due to expression of those core pathway components. And then the fourth mechanism is impaired systemic spreading of that silencing signal throughout the body to all tissues. And then the fifth mechanism is refractory target genes. So some genes cannot be suppressed due to their sequence and expression. So today we're going to talk about all of these in detail. And so let's go ahead and begin with instability of double-stranded RNA. You can see on the left here in lane 3 that double-stranded RNA is rapidly degraded in Lepidopteran, such as the European corn borer, but it's highly stable in Coleopterans, such as the western corn rootworm. And in a study where 37 different insect species were compared, double-stranded RNA stability was highly variable between species in Coleopterans and Hemipterans. However, double-stranded RNA was usually unstable in all the Lepidopterans investigated. So instability of double-stranded RNA is a widespread issue in insects. So what's causing this problem? Well, the leading enzyme that's been implicated is Dsrnases. So there's usually 1 to 4 Dsrnases in each insect species. In the case of the migratory locust shown here, there are 4 double-stranded RNase genes that are expressed in different tissues throughout the body. However, it's double-stranded RNase 2 that's actually lowering oral RNAi efficiency because the other enzymes are not compatible with the physiological pH where they're expressed, so they're not active. So interestingly, when groups have suppressed these double-stranded RNase genes in different species, such as the Colorado potato beetle shown here, they do get enhanced RNAi efficiency, and that can translate into improved plant protection against insect feeding. However, this isn't the case for all insects. In the case of the desert locust, suppressing the nucleases did not enhance RNAi efficiency. So another class of nuclease that's been implicated in Lepidopteran specifically is the REases. So these have mainly been implicated as lowering RNAi efficiency in the gut. However, recent studies show that these are predicted to be localized intracellularly, so they could be reducing RNAi efficiency in a variety of tissues. The really concerning thing about these is that they're upregulated a lot faster and more intensely than the core genes in the RNAi pathway, such as Dsrna2 and Argonaute2. And so that can definitely reduce RNAi efficiency. And when a knockout line of cotton bollworm was created that lacked the REase gene, you can see that it enhanced double-stranded RNA stability compared to the wild type. Unfortunately, RNAi efficiency was not enhanced because a plethora of other nuclease genes were upregulated upon knockout. So instability of double-stranded RNA is a major issue that needs to be overcome in insects. Moving on now to insufficient double-stranded RNA internalization. You can see here on the left that when double-stranded RNA is injected into fruit fly larvae, there's no gene silencing compared to the control. However, when RNA is expressed internally as a hairpin, you get efficient RNAi-mediated silencing. So this suggests that double-stranded RNA uptake is limiting in some species such as dipterans. So interestingly, internalization of double-stranded RNA works a little bit differently in insects than it does in a lot of other organisms where the SID transmembrane channels have been implicated. So in insects, there are SID1-like genes or SIL genes in some species. And in the case of like the Colorado potato beetle, they've implicated SIL-A and SIL-C in double-stranded RNA uptake. However, when you suppress both of those genes simultaneously, it only resulted in 24% rescue of the reporter. And so this indicates that SIL genes only play a very minor role in double-stranded RNA uptake. In insects, receptor-mediated clathrin-dependent endocytosis seems to be the main double-stranded RNA uptake mechanism. So you can see on the top left that double-stranded RNA localizes around the periphery of cells and shows up as this green ring or dots. But when scavenger receptors are knocked out, that signal is lost. So this implicates those receptors in double-stranded RNA uptake. Interestingly, in other species, researchers have used different inhibitors of different types of endocytosis. And in the red flower beetle, they were only able to implicate clathrin-dependent endocytosis in double-stranded RNA uptake. So one issue related to endocytosis that can be a problem in insects has been described in the oriental fruit fly. And these researchers showed that pre-exposure or pre-challenge with double-stranded RNA inhibited uptake of subsequent double-stranded RNA. However, naive cells could internalize double-stranded RNA just fine. So this issue needs to be investigated in other species, especially when RNAi of RNAi is going to be employed as an experimental technique, because pre-exposure could limit RNAi efficiency of subsequent double-stranded RNA. So a more widespread issue that really seems to impact a lot of insects is limited endosomal escape. So here on the top left, you can see that double-stranded RNA is internalized equally well in Lepidopteran and Collaborators cells. However, over time, double-stranded RNA remains in the endosomes of Lepidopteran cells, whereas in Collaborators cells, that double-stranded RNA is rapidly processed and removed from the endosomes. And you can see that even after 3 and 5 days after double-stranded RNA exposure, siRNA is only detected from the Colorado potato beetle tissues, and it's never detected from cotton bollworms, tobacco budworm, I'm sorry. And so there's actually a study in 37 different insects that showed that the lack of siRNA production is a problem in some species of hemipterans and orthopterans and many lepidopterans. So this is a widespread issue, and endosomal escape and uptake issues need to be overcome in insects to enhance RNAi efficiency. So now moving on to deficient core RNAi machinery. So this is a possible problem in a lot of insects, and in species such as Collaborators, there's actually been a lot of gene duplications in the core machinery of the siRNA pathway. However, there's not a very strong correlation between gene copy number and RNAi efficiency. And this is likely because unique components contribute in some lineages, such as the double-stranded RNA binding protein Stefan C, that's unique to Collaborators and is thought to enhance double-stranded RNA processing into siRNA. So to further complicate matters, the pathway components from multiple pathways, there's actually overlap between these pathways. So you can see here on the right that in the Colorado potato beetle, RNAi of RNAi completely blocked suppression of the reporter when they targeted Argonaut 2b, Argonaut 1, Argonaut 2a, and OBP. So this shows that different components of all three RNAi pathways can contribute to RNAi efficiency in some insects. Expression and upregulation of the core enzymes is also a very important part of this story. Here on the left, you can see expression profiles for Dsrna2, R2D2, and Argonaut2 throughout the different life stages of 4 different insects. And you can see here that expression of these enzymes is highly variable over time and between species. And so if you're trying to target a life stage that has very low expression of the core enzymes, you're likely to see lower RNAi efficiency than if you target other life stages or even other species with higher expression of the core enzymes. So another important component of this is upregulation of the core enzymes in response to double-stranded RNA. So there's work conducted in the pea aphid that suggests that low doses of double-stranded RNA are not effective in the pea aphid unless a high dose of non-specific double-stranded RNA is delivered first in order to upregulate those core enzymes. So another important factor that's been much less well studied in insects is functional between the core enzymes. So here you can see that there is variation in the domain architecture between Dsrna2 proteins and insects. In addition, if you look at key residues in the phosphate binding pocket of Argonaut 2 in insects, there's very low levels of conservation. And so these differences are likely translating into enzymatic or functional differences. Unfortunately, enzymatic kinetics have not been studied very well in insects. So far, there's only been one study that's looked at the biochemical kinetics of Argonaut 2 in fruit fly. And that study showed that insects likely has more efficient cleavage compared to mouse Argonaut 2. So it would be very interesting to see additional studies comparing enzymatic kinetics between different core enzymes from insects, because the function of these enzymes is going to directly impact RNAi efficiency. So now let's talk about impaired systemic spreading. This was first hypothesized to be an issue in insects because insects lack the RDRP enzyme that's responsible for amplification of the silencing signal in other organisms like worms. In addition, some insects lack SID orthologs, as we discussed previously. So there's been a lot of interest in what kind of mechanisms actually are responsible for systemic spreading of that silent signaling in insects. So studies conducted in Western corn rootworm suggest that there is no amplification of siRNA, and that indicates that amplification might not be a prerequisite for efficient RNAi silencing, since the Western corn rootworm has a very robust response. And interestingly, there's evidence from the Colorado potato beetle that suggests that long double-stranded RNA is the silencing signal. So above 60 base pairs, you can detect double-stranded RNA in the gut as well as in the carcass after oral delivery of double-stranded RNA. However, there's a lot of evidence from the fruit fly suggesting that amplification of siRNA does occur and that there can actually be multiple spreading mechanisms in insects. So on the left, you can see that there have been nanotubular-like structures observed that are transferring double-stranded RNA and Argonaut 2 enzymes between S2 cells. In addition, at least in the case of viral infections, double-stranded RNA is taken up by hemocytes, reverse transcribed into viral DNA for de novo synthesis of secondary siRNAs, which then spread throughout the body via exosomes. So this indicates that there can be multiple mechanisms of spreading and potentially amplification in insects. And really, we need to study the conditions under which these different mechanisms are active in different insects, because this is likely to influence RNAi efficiency quite strongly. So finally, let's talk about refractory target genes. So here on the left, you can see an example from the Asian corn borer that shows that when different genes were targeted with their corresponding double-stranded RNA, only some of them could be suppressed. There were also hormone and immune pathway-related genes that could not be suppressed despite extensive optimization. And when you look at RNAi efficiency for a single target gene, such as the chitin synthase A gene in these 4 species of Lepidopterans, you can see that the corresponding double-stranded RNAs resulted in very different levels of gene suppression in these different species. And it also resulted in different levels of mortality that were not necessarily correlated with the level of suppression that occurred. And so this could be responsible so target genes can be refractory in some species, but not in others. So what could be driving this? Well, differential dicing sites for different insects have been described recently. So if you look at a length of double-stranded RNA, you can see that there are some regions where a whole bunch of siRNA are generated, and then other regions where less siRNA are generated. And when you mutate the base pairs in those regions, you can actually change the abundance of the siRNAs. And so further research in Lepidopterans showed that GGU is the main dicing site in a lot of species. However, in the red flower beetle, dicing occurred at A's and U's, so there is actually a more diverse dicing sites in some species than others. So this could definitely influence within, between species and between gene susceptibility to RNAi. So another important component is the gene regulation that can also influence the susceptibility of different genes to RNAi. So again, in the Asian corn borer, researchers observed that genes could be put into 3 categories after double-stranded RNA exposure. So genes were either upregulated, unaffected, or downregulated by non-specific double-stranded RNA exposure. However, when these genes were individually targeted with their corresponding double-stranded RNAs, there was actually a subset of the upregulated and unaffected genes that could not be suppressed by their corresponding double-stranded RNAs, as shown in red and white. However, all of the downregulated genes, those could be knocked down by their corresponding double-stranded RNAs. So together, these studies are very important because they show both the sequence and gene regulation can have a major influence on gene susceptibility to RNAi. In addition, they offer tools that we can use to predict which genes are going to be good targets for RNAi. So to recapitulate, the stability, uptake, and endosomal escape are key issues that are limiting RNAi in insects. In addition, studies on biochemical kinetics of the core RNAi machinery and on mechanisms of systemic spreading are needed. Furthermore, investigation and screening of target genes is critical to success. So all of this information is important because a better understanding of mechanisms influencing RNAi efficiency in insects will facilitate strategies to overcome efficiency limitations so RNAi can be used in a wider range of insects. So great strides are being made in this field, and I encourage everyone to keep up the good work. Today, I only had the opportunity to talk about mechanisms limiting RNAi, but if you're interested in strategies for enhancing RNAi efficiency in insects, I direct you to our recent review paper that was published last month in Pest Management Science. So with that, I would just like to thank my co-authors for helping with this review, as well as our institution and our different funding sources. And I'd like to thank everybody from the iPlanta webinar for inviting me to speak with you today. And thank you all for participating. I look forward to our discussion later on. Thank you.
Thank you very much, Anastasia. I think you gave a very nice presentation, presenting a very good overview of some of the major barriers affecting RNAi efficacy. At the moment, I don't see any questions on the chat yet. We still have 2 minutes, so maybe I can ask you one question for now. I was wondering, you were mentioning about this amplification system that is present in nematodes, at least in C. elegans. Is there any clear evidence that there is definitely no amplification system in these highly sensitive insects? Or is it more a case of not finding the same system that is present in C. elegans, so probably there is no amplification system?
So it's a very good question. So the same mechanism that's present in nematodes has definitely been ruled out in insects. It seems to be a different mechanism completely. However, I'm not completely convinced that there's no amplification in species like Western corn rootworm. There are some technical limitations with some of the sequencing technologies that may have prevented detection of the siRNA amplification. So I don't know. I think we should definitely keep looking because that has not been resolved completely at this point.
Okay, thanks. Okay, maybe we can go to our next speaker. It's Dr. Jun-Sun Yoon. He's currently working at, sorry?
Yes, there is a question if you want.
Is there?
Was a chart where similar experiment conducted using, you can read it.
Yeah, so the question is whether similar experiments were conducted using siRNAs instead of Dsrna. I'm not sure which species this is concerning. Because so Dsrna is known to, well, for some species, it's known that long Dsrna is required, at least for cellular uptake. But I think there is also some evidence that for some species, for example, some hemipterans, they seem to respond to short interfering RNAs as well. So there could be some variability, I guess, between species there. I don't know, Anastasia, if you have anything to add here.
Yes, it seems like in most species, 60 to 100 base pairs is the limiting, the threshold for what will be taken through uptake mechanisms. However, now that nanoparticles and some other different coatings are being used, it seems like we can exploit different types of endocytosis or different types of uptake mechanisms. And in the case of species where siRNA works, it would be very interesting to look at the uptake mechanisms there and how they differ. Is it a different receptor mediating endocytosis? Is it a different mechanism completely? Are they relying more heavily on the SID transmembrane channels? We just don't know yet. So those are great avenues of future research.
Okay, thanks. So our next speaker is Dr. Jun-Sun Yoon. He's currently working at Oregon State University and will present recent work which he conducted while he was working at Cornell University on intraspecific variation of RNAi and aphids.
All right, can you guys hear me and see me?
Yes, we hear you perfectly.
All right, thank you for the introduction and thank you so much for having me here. This work has been done when I was in Cornell University, and as Olivia mentioned, I'm currently working at Oregon State University. Let's see. Yeah, this study has been published in IBMB last year in 2020 with the title Candidate Genetic Determinants of Intraspecific Variation in P. aphid Susceptibility to RNA Interference. So this study is largely divided into 3 parts. First, I'd like to talk about the natural variation in RNA susceptibility against alkyl-protein-1 dsRNA. And second, we believe that the determinants of RNA efficiency may be derived from the genetic background. So we perform the GWAS to find out which SNPs are affecting the RNA susceptibility. Then third, we select the candidate SNPs and validate those SNPs by RNAi of RNAi approach to find whether any of those SNPs are the determinants of RNAi efficiency. So here we collect the funded trig morpho from the field in Ithaca during this 2 weeks period in 2015. And out of 606 aphid lines, individuals, we select the 83 aphid genotypes for RNA experiment. So here are the process of collecting and selecting the aphid genotypes. Before I joined this group, Dr. Chung has been done tremendous work sorting out the unique aphid by microsatellite analysis, followed by genotyping by sequencing. So his paper used GBS to screen for secondary symbionts. And in my study, I randomly selected 83 lines out of 208 lines. And we believe that the 83 would be enough to represent the whole population, because it would be nice to do all 208 samples, but RNA susceptibility test was sort of screening test. So we use a lot of aphid. So here is the data. We orally delivered aquaporin Dsrna for 3 days, and water used as a control. And one of the left figures shows the relative survivor, which comes from this formula. And the aquaporin knockdown, aquaporin survivor over the water treatment. And as you see, there is a wide variation against aquaporin 1 Dsrna. And some of the lines are highly susceptible to aquaporin, which shows a 0% survivor. And some of them are not sensitive at all, showing 100% survivor or more survivor. On the figure on the right, except the ones showing 0%, we measure their body weights at day 3. And as you see, there is a wide variation on their body weight as well. And in order to see whether there is any correlation between low survivor and low body weight and high survivor and high body weight, we perform the correlation analysis. So the result says that the positive coefficient of 0.81 indicates a strong direct correlation between the survivor and the body weight. Then we want to make sure whether this RNA susceptibility is consistent with other lethalgens, such as snake skin, well known for membrane protein protein associated with the septic junction. So we select a few highly sensitive gene and highly sensitive aphid lines and non-sensitive aphid line from the previous result. This is aquaporin survivor. And then we performed the Dsrna feeding assay against SSK. So we have 4 highly susceptible and 4 non-susceptible lines here. And the purple dots are water and green dots are SSK knockdown. Let's look at the right figure first. Low susceptibility lines show also the low mortality, which is consistent with aquaporin 1 Dsrna treatment. However, on the left side, if you see the green dots, they are everywhere from 25 to 75. And here are 2 interesting facts. First, not all of them reach the 0% survivor, even though we collect the samples from the 0% survivor against aquaporin 1 Dsrna. And second, some of them are not even significantly different from water control, what it implies that RNA susceptibility of genotype is overlapping, but not identical for aquaporin 1 and SSK. We believe that the natural variation can be harnessed through the GWAS to study the genetic determinants of RNA efficacy. So we performed the genotyping by sequencing to get a SNP profile. Within 83 aphid genotypes, there were 141,000 SNPs. And using the nominal threshold of 10 to the minus 4, significant association were obtained for 103 SNPs. and which contains the 55 genes. And some of the enriched Gautam are shown here with those 55 genes. And among them, we choose 6 genes. These are the ACPY number that I'm going to use, and name of the gene, and brief function, and their class of generic variants. So you might be expecting to see the core RNA machinery gene here, but they're not shown in our GWAT study. And other than that, here are some 6 genes we selected based on their potential functions in RNAi. But the Trachellus was selected due to the lowest p-value, and the rest of them are based on the potential function in RNA pathway with the help of Gautams. So as you see the class of genetic variants, there are introns or even synonymous substitution. We don't expect these genetic variants to give a big phenotypical changes. And we wouldn't expect individual variants with a large phenotypic effects in natural population. But we expect the variation in RNA susceptibility to be a highly polygenic traits. So we move on to the validation study here. We validate the genes with RNAi of RNAi approach to see whether knockdown of this target gene, 6 genes affecting the RNA efficiency. We firstly knock down the nucleases for better RNA activity later. And we use the water as a negative control and GFP as another negative control, and aquaporin 1 as a positive control. There might be some mortality. And we want to see whether a gene of interest knockdown shows any mortality or to survivor to the aphid line. And what we want to know is, what if we mix aquaporin with a gene of interest Dsrna? Will they show any difference between the aquaporin 1 alone and aquaporin and gene of interest mix? So at day 8, we check the survivor, and if there is any difference in survivor between these 2 samples, the gene might have some impact on RNAi. So in this experiment, we firstly knock down these 3 genes. Trachellus and the nucleases, reverse transcriptase, and voltage ligand gate potassium channels. And this is how we get the relative survivor. And we use the post-hoc-done-net test for the statistics. So figure on the left, once we knock down the gene of interest, they don't really affect to the aphid survivor. And figure on the right, we used a susceptible line so that we can see the effect of candidate genes either go up or down. So aquaporin alone itself showing about the 60% survivorship. And compared to that, these 2 samples doesn't show much differences. But here, 1635, Trachellus was, survivor was rescued by 1635 knockdown, which is a Trachellus. And this result indicates that the Trachellus acts as an activator for RNAi, because once we lose the Trachellus gene, the RNA efficiency went down. And this is a second set of experiment trying these 3 genes with the same approach as previous slide. And knockdown of gene of interest doesn't show any differences. And in this aquaporin knockdown experiment, aquaporin control was less effective, despite the fact that the head case shows the significant difference. And 9356, pertrophin A was close to the significant level, and 9525 doesn't show the differences. So it seems like the head case and chitin binding pertrophin act as an activator of RNAi. So p-aphid genotypes collected from the single natural population vary in their susceptibility to RNAi against aquaporin 1. And what it means is there is a standing variation against aquaporin 1 Dsrna. And moreover, RNA susceptibility to the of genotypes is overlapping, but not identical between aquaporin 1 and SSK. And actually, our goal of this study was to find the suppressor of RNAi, so that by knocking down the suppressor gene, we can increase the RNA efficiency regardless of the genetic variation. So when I look back, this was an interesting approach to find the suppressor or activator using GWAS, which means a genetic variation as a clue to find the determinants of RNAi efficacy. So this project was supported by the USDA, and I really thanks to Angela Douglas and my lab members. And currently, thanks to my current PI, Manon Choi, now I'm working on spotted wing Drosophila, and I'm looking into various ways to control it with RNAi. And I am joining the Genolution company sometime in this April. So some of you already know about this company. They do Dsrna synthesis from milligram to kilogram scale. So if you plan to do some injection or a mass feeding assay, let me know. You can save your time and effort. Thank you so much for organizing this meeting, Bruno and Olivia. And thank you for listening, everyone. I'll take some questions. Thank you.
All right, thank you very much. We have a question on the chat from Professor Pali. He's asking, did you determine knockdown efficiency of the target gene? I'm not sure which target gene exactly. Maybe Professor Pali, you could.
Yeah, so most of the data seem to be on the mortality. I was wondering if that correlates with variability and knockdown efficiency. Did you look at the knockdown efficiency of these target genes?
We saw the knockdown efficiency, but it was not consistent. Some of them show the knockdown efficiency, but some of them are not. So we use mortality and then body weight as a phenotype as a RNAi effect.
Thank you.
Okay, one more question. Sorry, maybe I missed the point, but can you please explain why you wanted to target aquaporin 1 and snakeskin? Why are these genes so important in aphids? Oh,
yeah. Aquaporin is important for, especially for hemiapterans, because the osmosis balance is really important for those sucking insects. So we select the aquaporin 1, and then our previous publication proved that the aquaporin is important and it induced mortality to aphid. So we select the aquaporin 1, and SSK, I choose it for the personal reason, because the other paper also says that the SSK is highly expressed in the gut and it plays an important role. So I just choose the SSK for another lethal gene to test the RNAi susceptibility. Thank you.
All right, thanks. I think it's time we move to our next speaker. This is Professor Steve Wyatt. He will present his recent work conducted at his lab on alternatively structured Dsrnas and their different cellular uptake mechanisms. Steve, your microphone is off at the moment.
There we go. Now can you see? Okay.
Yes, we can see and hear.
Okay. All right, so you can see my screen?
Yep.
Yes? Okay. All right. First of all, thank you to the organizers for convening this meeting. It's proven to be pretty informative so far. I'm looking forward to hearing the rest of the talks. Today, what I'm going to talk about is my research on alternatively structured double-stranded RNAs and a different way that they may be entering insect cells. And for those people who attended the Entomological Society of America meeting, I do apologize, there's going to be some overlap, but there will be some new material coming. So hang in there. All right. My research is focused on applying RNAi for a few reasons that are probably similar to many other people. Firstly, we are interested in RNAi as a gene validation tool to create these transient loss of function phenotypes. In those cases, we often are simply injecting the double-stranded RNA into the insects to get a precise dose delivered to the insects. The other applications that we're interested in, which many of you are as well, is in the development of RNAi-based pesticides. In this particular case, we're looking at either making transgenic plants that express the double-stranded RNA, or we're looking at foliar sprays that we can apply to the leaves that the insects will ingest and get sick and die. And in both of those cases, again, we're looking at the insects ingesting the double-stranded RNA. We're also looking at ingestible double-stranded RNAs for the sterile insect technique, where we want to feed insects on double-stranded RNAs that will sterilize them and ideally sex sort them for sterile male release programs. And once again, in these kinds of applications, we're looking to feed them. So we're seeing ingestion as the primary route of entry. Now, thankfully, Anastasia has already described some of the aspects of uptake, so I won't belabor the point too much. What we see in insects is that there are a variety of ways by which the double-stranded RNA can enter the cells. Some insects have these SID-like proteins, but as we've heard, there seems to be some ambiguity about their role in uptake in some insects. There are a few cases of where phagocytosis has been observed to be involved in uptake, particularly where the double-stranded RNA is encapsulated in a microorganism, or where there are large macromolecular complexes where they're being used as carriers to bring the double-stranded RNA into the cell. But by and large, most insects are showing evidence of clathrin-mediated endocytosis, where clathrin is forming the vesicle comes into the cell, and the double-stranded RNA must escape the endosomal compartment to hit the cytoplasm to induce RNAi. Many people have studied this process using a variety of techniques. On the left, you can see that people can use fluorescently labeled double-stranded RNAs to visualize the double-stranded RNA within the cells, such as in the fall armyworm cells by Reddy Pali's research group. Or in the case of a fungal cell, which is from my lab, we can see uptake of double-stranded RNA within the fungal hyphae. We also will use chemical inhibitors to try to provide evidence for which type of endocytosis is occurring. And we can also use an RNAi-mediated approach to knock down key players of key proteins that would be involved in those processes to confirm which process is involved. Since my lab is working mostly on mosquitoes, I'm going to apologize, there won't be so much plant material here, but there will be some plant applications later in the talk. But since we're focused on mosquitoes, I used a mosquito cell line to first of all explore what uptake mechanism they use. And what you can see in the graph below is that what we were doing was treating the cells with these chemical inhibitors and then looking for knockdown of tubulin transcripts by applying the double-stranded RNA afterwards. And you can see on the left-hand side, there's the relative gene expression. And what you can see is that when we treat the cells with a clathrin-mediated inhibitor, like baflamycin or chlorpromazine, we are losing RNAi efficacy, which is telling us that clathrin-mediated endocytosis is the primary mechanism of uptake. What we see is that treatment with other inhibitors that inhibit phagocytosis, caveolin, or some scavenger receptors, we didn't see any impact on loss of RNAi, which tells us that they are not having a major role. Sorry, my slides seem to be on autoplay. But anyway, what we were interested in was exploring how many of these particular genes are actually proving to be associated with clathrin-mediated endocytosis. So we did the RNAi of RNAi approach. We treated the cells with the double-stranded RNAs targeting these various uptake cells. And then what we could see in this panel below is, oops, my apologies, is that we've targeted different genes associated with vesicle formation, with endosomes early or late. And when we inhibit any of the vesicle associated genes or the early endosome genes, we see no impact on RNA interference or of the second gene. So showing that clathrin-mediated endocytosis is in fact mediating uptake. And what we can see here is that treatment of the cells with a double-stranded RNA targeting a late endosomal gene, ARF72, is showing secondary knockdown, which tells us that the double-stranded RNA escaped the endosome prior to final maturation of the endosomes. Now, most of that's not terribly new. I think we all can appreciate that clathrin-mediated endocytosis is a likely mechanism for many insects. And what we have to at least recognize, though, is understanding uptake mechanisms is hugely important, particularly as we try to circumvent troubles that we're seeing with possible resistance in the field in the future, or with those insects that are naturally refractory, such as the Lepidopterans. So Anastasia nicely explained that we can see these various types of barriers to RNAi efficiency, such as gut nucleases, where people may be trying to either inhibit them or deliver the double-stranded RNAs with microcarriers that might protect them from the nucleases. Or we're looking at ways to get the double-stranded RNA to enter the cells when they may not. And this is a particular concern for those insects that are developing resistance, like the Colorado potato beetle and the corn rootworm, where they've been bred in captivity and are showing resistance due to uptake. So we were interested in trying to overcome some of these barriers. We took a slightly different approach, however. What we decided to do was look to see if we could protect the double-stranded RNA by changing its shape somewhat. So we were basically looking for, first of all, resistance against nucleases. Here, what we did is remove the guts from the insects, soak them overnight in a buffer, and then would treat that mixture with our different structured double-stranded RNAs and resolve them on a gel to look for degradation of the double-stranded RNA. What we can see here is that the short interfering RNAs and the long double-stranded RNAs are showing basically quick degradation over a 60-minute time period. The hairpin RNAs are somewhat more robust, and the paperclip RNA, which is this closed structure on either end, is showing considerable resistance to the double-stranded or to these nucleases that are within the gut. Here, what we wanted to test was whether these paperclip RNAs were behaving any differently than other double-stranded RNAs in their uptake. And we were surprised to discover that these paperclip RNAs are not entering the cells by clathrin-mediated endocytosis. This was based on these experiments where we treated the cells with chlorpromazine, which is an inhibitor of clathrin. And what we can see is here we have cells that are showing full or strong knockdown of the target gene when no chlorpromazine is present. But when chlorpromazine is present, the long double-stranded RNA is failing to enter the cells. The short interfering RNAs show only a small knockdown, but they too lose their efficacy when they're treated with chlorpromazine. And same with the short hairpins, they too are losing their ability to enter the cells and induce RNAi with chlorpromazine. But interestingly, the paperclips here are both showing ability to knock down the gene effectively, either in the presence or the absence of chlorpromazine, which tells us that these paperclip RNAs are entering the cells differently. We have now tested several different paperclips targeting different genes. And what we find is that in general, they behave better in terms of knockdown than the long double-stranded RNAs. Here you're seeing 3 different target genes, and the bars in red are showing that the paperclips are showing stronger knockdown than the long double-stranded RNA when the double-stranded RNA is not without chlorpromazine. So here we're seeing again, the paperclips are showing knockdown even in the presence of chlorpromazine. And in this panel, what we see is that the mortality is also greatly improved when the double-stranded RNA is delivered in a paperclip form relative to the long double-stranded RNA, where mortality rates are low but in all cases, these paperclips proved more effective at killing the insects than the long double-stranded RNAs. So one of the questions that we had was, how is these paperclips actually entering into the mosquitoes? Are they getting in by the gut lumen as we anticipated? To check this, we fed the mosquito larvae on double-stranded RNA that contained food coloring so that we could monitor the amount of food coloring that was going into their gut as a proxy to how much double-stranded RNA they were acquiring. What we did is then separated the mosquitoes into two groups. We had those that had a high level of dye in their gut, presumably eating a lot of double-stranded RNA, and those with a little or no dye in their gut, presumably eating very little double-stranded RNA. And in this graph, what we can see is that the sorting worked effectively, that what we see is that when we use densitometry to measure the intensity, we could see that the double-stranded RNA is accumulating in the gut in these highly pigmented guts, whereas there was only about 1/10 of the intensity in those that had little to no dye. In the bottom panel, we're looking at the knockdown of one particular gene that we were knocking down, SNF7. And what we could see is that we could get 58% knockdown in those insects that fed on a large amount of double-stranded RNA, and virtually no effective knockdown with those insects that ate very little. What was surprising is that with the hairpin molecules, while they showed a slightly better knockdown when there was lots of dye in the gut, they also showed a good deal of knockdown even when there was little to no dye in the gut. So that either tells us that these paperclip molecules are much more potent than we originally thought, or that they may be getting into the insects by some other mechanism. So we're actually going to explore that other mechanism by looking at topical applications to see if double-stranded RNA is actually the mode of entry, if it's actually getting in through cuticle or some other surface of the insect. All right, here comes at least some agricultural applications. The paperclip RNAs were tested on some other insects we have in the lab, which are flea beetles. Flea beetles are pests of canola. And so what we did was spray these leaves with different structured double-stranded RNAs, and then fed these paperclips, treated leaves to the insects. What you can see in the panel here is that the paperclip RNAs are generally showing stronger mortality than either of the other two structures that we tested, the long linear double-stranded RNA or a hairpin RNA. So once again, the paperclip is looking positive. Mind you, this is only one particular gene. We have many more to test to see if this trend holds true. We're also interested in applying these paperclips now to other organisms. And here we're testing it against a fungal pathogen. And I apologize that this is an insect audience, but I just want to illustrate that the same phenomena we're seeing with the paperclips in insects seems to apply in other organisms such as this fungus. Here we're looking at some pictures of Sclerotinia, which is a pathogenic fungus of canola. And what we could see was that we could label the double-stranded RNAs to accumulate within the hyphae. But when you treated these fungal cells with chlorpromazine, you would see that there would be restriction of the double-stranded RNA outside the hyphae. It was not entering the cells. And this was correlated with the knockdown. We could see that long double-stranded RNA, as long as there's no chlorpromazine present, we could get 45% knockdown. But when you add chlorpromazine, you don't get any knockdown. But once again, with the paperclip RNAs, we could see effective knockdown even in the presence or the absence of chlorpromazine. And I'm sorry, the auto slides are distracting. So you're probably asking, what is the mechanism of uptake of these paperclips? And the truth is that we still don't know for sure. We are conducting a range of experiments. We are labeling the paperclip RNAs to see that we can track them intracellularly. We're also looking at all of these various types of endocytoses to see which, if any, are associated with uptake of paperclips. And what you can see here is that we've ruled out clathrin-mediated endocytosis, flotillin, and calveoli-dependent endocytosis. But there are still other mechanisms that we're exploring to see which, if any of them, are relevant in uptake. And finally, I just want to summarize by pointing out that We recognize that uptake differences may vary, or there may be differences in uptake in different insects. And what we've at least found is that there's a paperclip molecule that, one, is resistant to nucleases, and two, it seems to enter the cells differently than conventional double-stranded RNAs. And why this might be particularly important is in cases where insects have developed resistance, we may be able to have another suite of molecules that could still deliver the double-stranded RNA to those resistant insects or to the refractory insects such as the Lepidopterans that are showing a lack of ability to allow the paperclips to be released from the endosomes. And finally, I'd just like to thank my team. This just shows some of the people that were involved in the insect research. And I'd be happy to take any of your questions. And I apologize for the speed at which these slides were rolling through.
All right, thank you, Steve. Great presentation. I don't know if there are any questions on the chat. Let's see. Marco is asking, how long were these paperclip RNAs you were using, and did you test different lengths?
Yes, all of the paperclips that we have been testing in these studies were short. They were only 23 nucleotides long. We are still questioning whether length matters, and those experiments are in progress at the moment. So I can't tell you yet whether a longer paperclip is going in by a different mechanism. It might be that the shorter size is what's enabling it to bypass conventional receptors and a clathrin-mediated process of uptake.
Okay, so the pathways that you've ruled out, you may want to rule them back in once you try with long Dsrna, for example, or long paperclip RNA, for example. Yeah. Okay.
Yes. Yeah.
Maybe one very tiny, but there's another question. Maybe I missed it, but do you have any data from Lepidoptera or stingbugs on stability, uptake, efficiency?
Yes, we do have data on the application of paperclips to Lepidopteran cells, and it does appear that it will induce RNAi. So that's intriguing because it might tell us that these paperclips are indeed bypassing some of those endosomal compartments. I didn't present those data here because the students are still trying to gather all those data.
All right, we'll be looking forward to that. I had a small question. How do you prepare the paperclip RNAs? I don't know if I may have missed it, but.
Oh, I didn't describe it. That's quite all right. We've just taken a couple of different approaches, but it can be done by in vitro transcription. You just simply have to add a T7 promoter in front of this, and it will assemble a fold over on itself. So it can be one contiguous transcriptional unit, which means that it would be applicable to, say, a transgenic approach for plants.
All right. All right, thanks. One more question, final one then. Do you think there are any yet undiscovered non-classical mechanisms of endocytosis in insects that could be involved in Dsrna uptake?
Well, on one hand, I kind of hope so, because it gives us lots more things to explore. If I rule out those other methods of uptake, then I will most certainly be looking for alternative methods of entry. I guess what we've noticed is that there are some small RNAs, examples of small RNAs that seem to be able to penetrate tissues more readily. And that's been applied to plants and insects. So it's possible that maybe these small molecules can behave differently and may bypass conventional endocytosis processes.
Okay, we're running a little bit over time now. So the questions, keep them firing. I will store them all for the discussion at the end. So our next speaker is Dr. Geert Plating from Syngenta, from the Ghent Innovation Center, previously DefGen. And he will give a presentation on delivery of Dsrna to plant tissues with the aim of controlling insects.
Thank you, Olivier. Let me share my screen.
Yeah.
Okay, there we are. So First of all, I would like to thank the organizers, Olivier, Bruno, for giving me the opportunity to give this presentation. My name is Geert Plating, and as Olivier said, I work at the Syngenta Innovation Center in Ghent, Belgium. And today I would like to share with you some insights we got into the mechanism, the possibility of delivering double-stranded RNA to plants. Okay, the next slide. So maybe first an introduction. So in the Ghent Innovation Center, we are developing RNA-based biocontrols, where we use topical application of double-stranded RNA for insect control. I don't think I need to explain to this audience how RNAi works. The attractiveness of this is that it has a unique mode of action, possibility for high selectivity of your products that you can design, and it's also anticipated that there is a low environmental impact. So the research focus we have in Ghent is on different insect orders like leaf-eating beetles, sucking pests, sting bugs, mites, leps, and then some soil pests. And all these have their different challenges and hurdles. Some have a very robust response, like some coleops, like Colorado potato beetle, corn rootworm. Others have other difficulties, and some are pretty recalcitrant to RNAi, like the labs, like it was already discussed. Okay, the next slide. So what could be the importance of implanted delivery for insect control? Well, first of all, the only way so far demonstrated that insects can take up double-stranded RNA is through oral uptake. So they have to feed on a substrate that contains the double-stranded RNA. And if we think about some pests that, like sucking insects or mining insects that rather eat from inside the plant, it's very difficult to make double-stranded RNA accessible to these insects. Also, if we could bring in the double-stranded RNA in a plant, maybe we could have a kind of a systemic spread of the active ingredient through the plant vascular system. Now, how could you bring double-stranded RNA into a plant? Well, the first method is through in-planta expression of double-stranded RNA, an RNAi trait, and you all know about the RNAi trait in corn against corn rootworm. But of course, this is a GMO trait, and it has its issues. Also, the development and the registration costs for a GMO trait are quite high. So this is an approach that is only valuable for, let's say, high-value, high-volume row crops. What you have in a transgenic plant is that you have a continuous production of double-stranded RNA in the plant. So a new supply of double-stranded RNA all the time. But the expression of double-stranded RNA might just be insufficient. And we have some examples in the lab where we cannot reach a high enough level of double-stranded RNA to trigger a response in the feeding insects. Also, you cannot really help the active ingredient through formulation. Now, if you want to use a topical application of double-stranded RNA as a spray, for example, as a bioinsecticide, there is really a need for an efficient production line of double-stranded RNA, which should be upscalable, cost-effective, and of course in line with the regulatory requirements. An advantage of this is that you can help actually uptake of double-stranded RNA by using the appropriate formulation. So you can help update both in the plants, but also in the insects. You can use different applications, technologies, spray, seed treatment, root drench, what you name it. And also compared to a GMO trait, you have a better control of timing and dose of application of double-stranded RNA. And also we think that this kind of technology has a broader application range and you can use it on smaller niche markets. Okay, what are the different obstacles and challenges if you want to bring double-stranded RNA inside a plant? One of the first and serious roadblocks is the cuticle, the plant cuticle. So can you penetrate the double-stranded RNA through the plant cuticle? Or can it be taken up, for example, by the stoma? And when you succeed in bringing double-stranded RNA in the plant, what about the stability? We know that double-stranded RNA in certain environments is quite unstable. Like for example, in soil, it's very rapidly degraded by all kinds of RNAs. So what about the stability of double-stranded RNA inside the plant? And then you have the issue of the systemic spreading of the double-stranded RNA. Can you get it into the circulatory system? And is it spread via the xylem or the phloem or both? And then finally, if you want to bring the double-stranded RNA inside the plant cell, you have to cross the cell wall and the cell membrane. So it's quite a challenge to bring biologically active double-stranded RNA inside the plant tissue. So one of the first things that we tried a couple of years ago already is to deliver double-stranded RNA into plants via foliar application. And the aim was to trigger an RNAi phenotype inside the plant. So this was inspired by the Bayer Monsanto BioDirect technology, where they showed that they could restore herbicide tolerance in resistant weeds by knocking out the ESPSP gene. So we tried, and to make a long story short, we tried very different approaches like physical wounding. We used all kinds of co-formulants, adjuvants, spreaders to get it inside the plant, biolistics, even this kind of specialized spraying nozzle, the Vortec technology, where you have a high speed, let's say, high-speed spray of your double-stranded RNA on the plant tissue. And so as I said, we tried many different things, but we were unable to see any evidence for effective foliar delivery of double-stranded RNA. So we took a more systematic approach than we were looking where the barriers were. And so one of the first barriers that we were looking at was the cuticle. And our colleagues in Stein, Switzerland have this kind of nice cuticle transmigration assay, which they regularly use to study actually penetration of chemicals or biologicals through a plant cuticle. And essentially the setup is as follows. So you have an upper chamber, which is the receiver solution. And then you have your active ingredient or whatever you want to measure, which is applied in this kind of controlled circulatory system and is allowed to get to penetrate through the cuticle. So we tried this approach to see if we could push double-stranded RNA through the cuticle, and we tried both simple double-stranded RNA or double-stranded RNA that was complexed or that was in combination with all kinds of formulations. And the detection methods that we use to see if any double-stranded RNA came through was HPLC, or we also used fluorescent labeled double-stranded RNA, or we even looked at double-stranded RNA in a bioassay. As you know, the Colorado potato beetle bioassay is a very sensitive assay. I will come to that later. But again, here we had no evidence whatsoever that double-stranded RNA, long double-stranded RNA, to make it clear, was unable to cross an intact cuticle. Okay. The next thing that we looked at was if plant cells could take up double-stranded RNA. Here we used fluorescent labeled double-stranded RNA. And if you just take, let's say, double-stranded RNA only, we could not see any uptake of double-stranded RNA in the plant cells. However, if you combine the double-stranded RNA with some uptake enhancers, like, for example, silhouette or a cell-penetrating peptide or polyethylenimine, we could see that double-stranded RNA could get inside the plant cell. Next, we were also looking if this kind of double-stranded RNA that we could get inside the cell could trigger a plant RNAi response. And for that, we used an Arabidopsis line or Arabidopsis lines that expressed a luciferase gene. And we supplied double-stranded RNA to these cell lines with the aim to knock down expression of the luciferase. And as you can see here, if you just take naked double-stranded RNA, you don't see any effect. We were unable to knock down the luciferase expression in these cell lines. However, if you combine the double-stranded RNA with polyethylenimine, you see an effect, but only at high doses. So yes, you can bring double-stranded RNA inside the plant cell, but it's not a very efficient process, and you need high dose of double-stranded RNA. Okay, but the question was, and this brings me back to actually the topic of my speech, can you get double-stranded RNA inside a plant, and is this double-stranded RNA still able to elicit a phenotypic response in the feeding insects? So for this, we used this kind of setup where you have a cut potato leaf that was put in a container containing double-stranded RNA and some dye. So these leaves were allowed to soak up the liquid, and we could follow uptake of the liquid with the dye. And after 24 hours, we took these leaves and we cut out some leaf discs that were fed to the Colorado potato beetle for scoring an effect. What is important is that there was never physical contact between the double-stranded RNA and the surface of the leaf here. So whatever the insects got was inside the leaf. And as you can see here on the right-hand side that, yes, you can really, let's say, deliver by this kind of method biologically active double-stranded RNA in the plant tissue and at a high enough dose to really kill a feeding insect, as you can see here. As control, we used double-stranded RNA against GFP or dye only. And so it was clear that through capillary uptake, you can move double-stranded RNA inside the plant to sufficient amounts to have an effect in feeding larvae. So we thought that was an interesting observation, and we looked at this more systematically using this kind of hydroponic tomato model linked to a Colorado potato beetle readout. So we have these this kind of hydroponic setups where we use cut stem plants that are soaked in this kind of solution. We also used cut root plants or intact plants. So they were incubated in solutions with double-stranded RNA. And then we scored for activity in the leaf disc assay, as I described before. And we also looked biochemically by quantigene. We tried to quantify the amount of double-stranded RNA that was present in the plant. So and these are some of the results. So here, this is a cut stem type of approach. So this is a cut stem plant that was put in liquid, and then we, after some time, we harvested the lower leaves, the middle leaves, the upper leaves, and you can see that at different time points you see accumulation of double-stranded RNA in these plant tissues. And what was also clear is that, let's say, accumulation was more efficient in newly grown leaves, in younger leaves versus older leaves. So this is this we could see in this kind of experiment. Okay. So the next question was, what is happening with this double-stranded RNA? How long does it stay active in the plant after this way of application? And what we did here is we soaked this kind of cut plant parts in double-stranded RNA, and after 20 hours, they were transferred to a solution without double-stranded RNA, and they were kept for longer time. And then we scored biological activity in different leaf parts of the plant. And to our surprise, this double-stranded RNA was quite stable in these plants. So this is after 7 days, and after 14 days, we can still see activity when you feed these plants to Colorado potato beetle, which is quite remarkable. Another interesting finding was that, so during the course of these experiments, these plants developed new tissues, new leaves. And if you look into these newly formed leaves, this is the yellow line here, we cannot find any activity. So some way this double-stranded RNA is taken up by the plant. It is stored in this kind of plant tissues. It remains stable for quite some time, but it is not transported back to other tissues. I think that was quite clear from this experiment. We also had some evidence, or we also think that actually the double-stranded RNA is taken up by the xylem and does not end up in the phloem, but we don't have any conclusive experiments to prove that. So nevertheless, we thought that this could be an interesting approach for, let's say, crops or fruits, vegetables that are grown in hydroponic setups, such as tomatoes and strawberries. And the idea was, if you could, for example, apply double-stranded RNA to the hydroponic medium, maybe you could, bring the double-stranded RNA in the plant and protect the plant against some feeding insects. So in this kind of hydroponic setup, we had two approaches. Either we took the intact roots, or we just clipped parts of the roots and put them back in the hydroponic solution. And whenever you use cut roots, like we see for cut stems, you have efficient uptake of the double-stranded RNA. You can see biological activity. However, if you use intact roots, it's not that efficient. Another thing that we tried was actually a soil drench approach, where we just almost saturated the soil. So this was not in hydroponics, but in soil. We saturated the soil with a high dose of double-stranded RNA in solution, and look if some was taken up by the plants, but we could never see any, or we could not measure any effect in the feeding assay. So the conclusion of this experiment was that there is uptake of double-stranded RNA via the cut roots, but probably not by intact roots. And this brings me to the conclusion slide. So as I said, we tried many approaches, but we found no evidence that there was effective foliar uptake of long double-stranded RNA in plants. And I want to stress that we used the regular long double-stranded RNA. We did not try modified RNAs or short RNAs, siRNAs or miRNAs. The situation might be different there. So we tried to push it through the cuticle. We were able to do that. In a cell line, we could deliver double-stranded RNA that triggered an RNAi response in the cell line. In this kind of hydroponic experiment, we showed that in cut plants, there is an efficient uptake and spreading of double-stranded RNA in the plant tissue, which is more intensive in younger tissues versus older tissues. We also see a very good stability of the store of the RNA that was taken up. But we don't see any redistribution once it is inside the plant to newly formed leaves. And also in the roots, you can trigger RNA, double-strand RNA uptake if you clip some roots, but with intact roots, it's not efficient. So, and with this last slide, I would like to thank my colleagues who contributed one way or another to this work. So our colleagues in Ghent, Belgium, Duncan, Nathalie, Annelies, Kimberly. Then Anke Buchholz in Syngenta Stein did this kind of nice cuticular transmigration assays, and our colleagues in Jallis Hill, UK, Rich, Sarah, Martha, Rachel, and Carl. And that's my last slide. Thank you very much.
Okay, thank you very much, Geert, for the great presentation. I think we have time maybe for one question. I see 2 questions on the chat, 3 questions. Maybe the first one, do you think Dsrna delivery by tree trunk injection, I don't know if it is feasible or if you have any experience with this maybe?
We don't have any experience with it, but I think it would be an interesting approach to look at. Of course, these are not simple experiments, and maybe also need a high dose of double-stranded RNA, I don't know, but it's certainly worthwhile looking at, I would say, for some whatever bark beating beetles or something like that.
Yeah, maybe one more short question. What is the potential of controlling phloem feeders, you think,
well,
with using exogenous application of Dsrna?
Well, as I said, probably for phloem feeders like aphids, you have to bring the active ingredient inside the phloem, which is very difficult through topical application. With this kind of hydroponic application, we never tried aphids. I think it's one of the things that we could try, but we don't have any data on that. But I said our fear is that it's not in the phloem, but it remains within the xylem.
Okay, we have several more questions, but I will keep them for the discussion at the end because we're already a few minutes over time. So thank you, Geert, again.
Yeah, I stopped sharing. Yeah.
So our next speaker is Professor Reddy Pali, who is from the University of Kentucky. And he will present his work on his recent work on improving RNAi in Lepidopteran.
Okay, do you all see the one presentation mode?
Yes, we do.
Thank you. Thank you, Oliver, for the invitation, and thank you all for attending. I'm going to try to give you some of the latest developments in my lab on improving RNAi in Lepidopteran insects. As you have seen from previous presentations, that there are several hurdles for efficient RNAi in Lepidopteran insects, and anesthesia nicely summarized 5 of those. In my presentation, I'm going to concentrate on 3 of those mechanisms listed on this slide. And I will use some of the data from my lab to show how we think that the top 2 are the main reasons, and the third one is probably not an underlying mechanism for inefficient RNAi in insects, and we can overcome using some of the approaches like nanotechnology. So, as you have seen from previous presentations, degradation of dsRNA by dsRNases and accumulation in the endosomes contribute to inefficient RNAi in fall armyworm. In the next couple of slides, I'll show you some of the work we have done in fall armyworm to show indeed in this insect also that is the case, both in degrading RNA by dsRNases as well as its accumulation in the endosomes. This is one of the early papers published by Sunny, showing that the dsRNAs are less efficient in degrading dsRNA both in the gut and the hemolymph compared to Lepidopteran insects, where the dsRNAs are highly efficient and they degrade dsRNA. So this seemed to be one of the major hurdles for efficient RNAi, at least in fall armyworm and other Lepidopteran insects. The second one is this intracellular transport of dsRNA, where dsRNA seem to get into the cells but get trapped in the endosome. That's what is shown in this slide here. These the green dots are the late endosomes, and the red dots are the endosomes accumulating dsRNA, labeled dsRNA. And when we match those yellow spots are the ones that are late endosomes accumulating dsRNA. So through these studies, we showed that these two, degradation of dsRNAs by dsRNases and accumulation in endosomes significantly contribute to inefficient RNAi in Lepidopteran insects. Now, the next question we want to address is, are there any differences in the expression of RNAi machinery that contributes to this inefficient RNAi in Lepidopteran insects? To address this question, we turn to this SID1 protein discovered in C. elegans. In C. elegans, these mutants of this systemic RNA interference deficient protein SID1, when they feed double-stranded RNA targeting GFP gene to these, the RNAi is very, very inefficient. Through a series of studies, this group showed that this is because of the protein that makes a specific dsRNA channel to take up dsRNA into cells. And this channel, the uptake is passive. That is what is shown in these bar diagrams on the left. They can, the energy is not needed, and it can happen at various temperatures, including 4 degrees. And by recordings, they showed that this particular channel is dsRNA specific. dsRNA, you can get the conductance, whereas with the double-stranded DNA, we don't get much conductance. So this SID1 seemed to mediate dsRNA uptake through passive diffusion. Several labs have tried to find homologs of the SID1 in insects, and there are genes that have similar sequences as SID1. They are some of them are labeled as SID1-like proteins. So, for example, in Western cornroot, SIL-A and SIL-C were reported. So, as in Colorado potato beetle and other insects. But whether they really function as double-stranded RNA channels is questionable. There are some silencing experiments where you knock out one of these SID1-like proteins and show some decrease in RNAi efficiency, like what is done in Colorado potato beetle and other insects, especially like in this brown plant hopper. Su et al. Published data showing that this is absolutely required. Without this molecule, RNAi doesn't work, but there are also some partial requirements. In Lepidopteran insects, it's not clear whether SID1-like proteins play any role in RNAi efficiency. There were some experiments done in cell lines showing that if we express these molecules in cell, and we can improve RNAi. In one experiment by Kobayashi et al. In silkworm, they tried to make transgenic silkworm expressing SID1, the nematode SID1. They did not see any improvement in RNAi, so it's kind of variable. It works a little bit in cell lines, but in vivo, at least in one try, it didn't work very well. We express this SID1 from C. elegans in Spodoptera cells, both ovary-derived SF9 cells and midgut-derived SF17 cells. As you can see on the top pictures here, when these cells, if we express SID1 in SF9 cells and hit those cells with Dsrna targeting inhibitor of apoptosis, which promotes apoptosis and cells die, you can begin to see some cells undergoing apoptosis in the bottom panel here. But if we do the same experiment in midgut-derived SF17 cells, expression of SID1 does not help RNAi at all, no apoptosis. And when we look at the luciferase activity in cells, we made stable cells of these SF9 and SF17 expressing luciferase, and when we expose them to Dsrna targeting the luciferase gene, we can knock down the luciferase gene in the presence of SID1 very efficiently in SF9 cells, but not in midgut cell line. Interestingly, the sensitivity of these cells to Dsrna increases tremendously. We can see even with 1.68 nanograms, we begin to see knockdown, whereas in the wild type cells without SID1, there is hardly any induction of RNAi. This we got intrigued, so there seemed to be difference in how the transport of dsRNA occurs in these cells. We wanted to make transgenic polar muon and see how different tissues respond to this SID1 in improving the RNAi. This slide is showing where when we express SID1 in SF9 cells, the accumulation of the dsRNA in these cells goes down. That is the graph here, about 50%. And we don't see processing of dsRNA to siRNA in normal SF9 cells, but once we express SID1, we can begin to see the processing of dsRNA to siRNA, showing that RNAi is working when we have SID1 expressed in these cells. To see these differences in vivo, we express this SID1 in polar muon by making transgenic insects using piggyback system. We used GFP driven by IE1 to screen the transgenic events where SID1 is expressed under IE1 promoter. Using this green color, we can pick out transgenic insects, and when we dissect them, we see quite a bit variation in the expression of this GFP in various tissues. This may be due to the activity of this promoter that we use to express GFP. And these GFP positive animals show the SID1 RNA suggesting that these transgenic animals do turn on this gene and make mRNA. We quantified the levels of both GFP and SID1 in various tissues, as shown in the top panels here, in the epidermis, fat body, midgut, Malpighian tubules, salivary glands, and so on. And most of them express high levels of GFP as well as SID1. One particular tissue we saw quite a bit of expression of both these proteins is Western's glands. Western's glands are the modified epidermal cells in insects. They make cuticular lipids and all involved in the synthesis of new integument when the insects molt. These are the green dots in these larvae are the Verson's glands. When we can dissect and we confirm them by various microscopy techniques. If we take these larvae, either wild type or SID1 expressing larvae, and treat them with either DS-Luc as a control or DS-GFP to target GFP. The wild type larvae, there is no detectable knockdown of GFP. But in the SID1 transgenic line larvae, when we treat them with GFP, we can see disappearance of GFP from these Verson's glands and other tissues, whereas when we treat them with the control Dsrna, we don't see that. And when we quantify knockdown efficiency by measuring GFP mRNA levels in these insects, we saw quite a bit of variability in different tissues, epidermis, fat body, all the way to Verson's gland. The highest knockdown efficiency we saw was in Verson's glands, almost more than 90%, whereas tissues like midgut and salivary gland, there is hardly any knockdown. So even though the SID1 is expressed in some of these tissues like midgut, still it is not able to help with RNAi in these tissues. In Verson's glands, we can not only knock down the GFP as a marker, we can also knock down endogenous genes like IAP. So this is a control panel where these SID1 transgenic larvae are treated with DS-Luc, a non-specific dsRNA. If we treat with GFPSP, the GFP completely disappears, the GFP. And if we treat these with the IAP, it induces cell death. The size of this Verson's gland decreases significantly, showing that the RNAi works well and we are able to get rid of a lot of cells from these glands. So what this research showed us is the tissues and cells of Lepidopteran insects contain highly efficient RNAi machinery, especially cells like ovary-derived SF9 cell line or Verson's gland cells in the larvae. But yet the RNAi is inefficient if we use naked dsRNA. That may be not due to a difference in the expression of RNAi machinery. It may be due to these two, degradation and accumulation, and getting enough of these molecules into the cytoplasm of the animal cells. There are various ways double-stranded RNA can enter the cell. You have seen that naked double-stranded RNA, several studies have shown that it is through clathrin-mediated endocytosis. It might be, some of it might be released into the cytoplasm, but most of it might be accumulated in endosomes. But there are other mechanisms like SID, like channels, through some kind of a fusion, like phagocytosis and other fusion-mediated entry. Or we could try to formulate these double-stranded RNAs to help them get into the cells, but either efficiently escape endosomes or bypass these pathways, go directly into the cytoplasm. That's where we think that we will be able to improve RNAi well in these Lepidopteran insects. The current focus of my lab is trying to find ways to bypass this endosomal accumulation of double-stranded RNA by delivering these dsRNA using different nano formulations. And we have developed a system where we can employ a battery of assays like Luciferase assays, pharmacological inhibitors, and apoptosis induction, mRNA levels and bioassays to screen number of nanoformulation, trying to identify the ones that efficiently deliver double-stranded RNA to kill these Lepidopteran insects. I don't have time to go into all that, but we have some nanoformulations in larval bioassays. We do these mostly in neonate larvae. We can achieve 60 to 80% mortality, and we begin to work on those, and the goal is to reach 100% in these assays, so we can try to take these to the next level. That's where we are. I'm going to stop there and summarize, telling you that the major reasons for inefficient RNAi in fall armyworm and other Lepidopteran insects are degradation by dsRnases and accumulation in the endosomes. Probably not differences in the expression of RNAi machinery. There is RNAi machinery in these cells. If we are able to deliver them to the cytoplasm, we'll be able to achieve efficient RNAi in these cells. And we think that nanoformulations might help in this respect. I would like to thank you all for attending this talk. And these three two postdocs and graduate student, Chan and Dandapani and Jinmo, are the ones did most of the work I presented today. And this work is supported by CAMTech and USDA through NIFA grant and Hatch grant. Thank you all very much.
Thank you very much for the presentation. Since we're a little bit over time, I'll just ask, there's one technical question from James Baum. He was wondering whether in the SID1 fall armyworm tests, whether you were using feeding or injecting Dsrnas.
Both. We tried both feeding and injections. And most of the data I showed is by injection. It's more efficient. But we also have some evidence that by feeding also we can knock down genes, at least in Verson's glands, not in other tissues.
Okay, thanks. We'll address the other questions afterwards. So our next speaker is Juan Luis Jorafuentes. He's a professor at the University of Tennessee, and he will present his work on the potential for resistance emergence in populations of Colorado potato beetle exposed to insecticidal Dsrna.
Hello, and thank you, Olivier, for the invitation to participate in this webinar. So as Olivier was indicating, in my presentation today, I'm going to give you an overview of our research looking at resistance to double-stranded RNA. But before I start, I would like to acknowledge my co-authors. First, Swati Mishra, who is the awesome graduate student that is pictured right here in the center, holding a Colorado potato beetle larva, who has done most of the research that I'm going to be sharing with you today. Jim D, which did most of the heavy lifting initially in selecting for resistance in Colorado potato beetle, as well as Bill Moore, who from Bayer Crop Sciences, who has had significant contributions to our research. So as you have heard today, there is a substantial interest in developing double-stranded RNA as a pesticide to control insect pests. There's a lot of research that has been ongoing to try to optimize this technology. So it's high specificity, represents a new mode of action, so it's a very interesting technology to develop for pest control. It also allows its use in both a transgenic and non-transgenic delivery. And so you have here a transgenic plant producing the double-stranded RNA versus what it would be a foliar spray of double-stranded RNA. I'm not going to go into the different steps of the mode of action of double-stranded RNA because it has been explained by previous presenters. But I wanted to make the point that, at least in theory, an alteration in any of these steps could potentially lead to resistance. In contrast to all that we know about how double-stranded RNA and RNAi works, we don't know a lot about what mechanisms of resistance insects may develop against these pesticides. In fact, the only published report of resistance to double-stranded RNA is this outstanding publication by Kajuri et al., in which they selected for resistance to a transgenic corn producing double-stranded RNA targeting the SNF7 gene in the Brodica bilgifera, the corn rootworm. So this selection resulted in about 130-fold resistance, which was autosomal, recessive, linked to a single locus, and it was associated to reduce uptake of the double-stranded RNA, as is shown in this picture here, comparing uptake of fluorescently labeled double-stranded RNA in susceptible with resistant insects. These resistant insects were also cross-resistant to alternative Dsrna targets, as shown in this graph, all these different gene targets, but they were not cross-resistant to the Cry3BB1 protein from Bacillus thuringiensis, which is expressed in transgenic corn to control these rootworm larvae. This was selection for resistance to a double-stranded RNA that was delivered by a transgenic plant. In contrast, there is no information on what resistance would look like when the double-stranded RNA is delivered using a non-transgenic approach. For this reason, we started our project using Colorado potato beetle, Leptinotarsa decimilineata, or CPB for short, and a topical application of double-stranded RNA. Why use this system? There's a number of reasons, but the first one is that this insect, one, is a very economically relevant pest of potato and other solanaceous crops that is highly notorious for its ability to evolve resistance to different pesticides, even different modes of action. It is highly susceptible to double-stranded RNA, so there's potential for developing this technology to control this pest. And in fact, it's the current target of double-stranded RNA pesticides that are being either developing or are already in the registration phase. So this increases the importance of research to study the mechanisms or the potential for resistance in this insect to topical double-stranded RNA. So to start our project, we first decided to try to generate what we call the general colony or GC colony, which was going to be our starting point. And we wanted this colony to be as genetically diverse as possible to increase the probability of having some major resistance genes in our population that could be selected. For this, we recruited the help of a number of collaborators in 9 different states in the US. So they provided collections of larvae and adults from the western, northern, and eastern population range of the Colorado potato beetle in the United States. Once this GC colony was generated by pulling all these insects that were collected from the field, we then developed a bioassay protocol that was also used for selection, because we were interested in identifying resistance alleles that were relevant for field use of this technology. We decided to start our protocol using second instar larvae that were exposed to potato plant leaf that was coated with a solution of double-stranded RNA. In this particular case, it was double-stranded RNA targeting the VATPA subunit A, which is highly active against Colorado potato beetle. So these larvae would grow feeding on these leaves coated on the double-stranded RNA. They would be changed to bigger containers as they grew in size. And around day 9 or 10, they would be moved as pre-pupa to soil containers where they would undergo pupation. and then a close at day 20 as adults. And what we think it was important in our protocol is that we use this emergence of adults as our endpoint, rather than mortality at the larval or pupal stage, to really determine the toxicity of this double-stranded RNA. So this table shows you a summary of the initial selection episodes. We started with around 300 individuals from the general colony, and 300 individuals that we had used from the general colony to perform initial bioassays to determine the LC95 dose of this double-stranded RNA, which was 0.38 microns per mil. That was used as the initial selection dose. So these individuals that survived these initial bioassays were pulled into a subcolony, and we used 300 individuals from this subcolony. The only survivor from the general colony and the 11 survivors from this adult survivor colony were pooled to generate the chronic exposure adult survivor, or CS colony. We took the CS colony to an F2, so we did 2 generations of seed mating to be able to obtain a higher number of individuals and then reselect with the original double-stranded RNA dose. And as you can see here, we had a fairly good number of adult survivors already at this stage. We then basically collected the adults that survived this exposure to generate another CS subcolony that was exposed to an increased amount of double-stranded RNA. So using this foliar exposure, it really allowed us to increase the amount of double-stranded RNA that these resistant insects were exposed to in different episodes of selection. In some of these episodes of selection, we also ran a bioassay with the GC parental colony to be able to see that the double-stranded RNA was active. And so these survivors in the adults were really representing resistant individuals. And we reached all the way to selection with 300 microns per mil of double-stranded RNA. And at this stage, we observed 95% survivals in adults and no survivors in the GC colony. So we took all this, the colony resulting from this selection to 300 microns per mil, and we performed a dose bioassay. And from this dose bioassay, we obtained that for the GC colony, the LC50 was 0.18 microns per mil, but for this CS colony, there was no significant mortality even at 2 milligrams per mil. So that supports that this strain has more than 11,000-fold resistance to this double-stranded RNA targeting the VATPA subunit A gene. Suari performed characterization of the genetics of this resistance. In here, you just have a table displaying the mortality observed in a single dose assay, in which we have crosses between the susceptible and the resistant strain. What you can observe is that in the heterozygotes, basically when we cross susceptible and resistant insects, the susceptibility is similar, if not identical, to the susceptible parents. And it doesn't alter whether we use a female resistant or a male resistant individual in the cross. So what this supports is that resistance is not sex-linked and is transmitted as a recessive trait. Suari has also performed substantial back crosses and bioassays with the offspring from these back crosses to determine that resistance in this strain is polygenic. I'm not able to share this data here because of time constraints, but she has done this work. One thing that we looked at was a cross resistance to additional double-stranded RNAs. And for this, we use double-stranded RNA targeting the coatomer subunit B beta gene. So as you can see here, in the resistant strain shown in blue, there's no susceptibility to either the VATPase target or the coatomer subunit beta target, compared to the parental susceptible strain or another susceptible strain called the MET strain that was provided by Andre Aloykin. So these insects were cross-resistant to an additional double-stranded RNA target, but we also were interested in testing susceptibility to the Cry3AA toxin from Bacillus thuringiensis because it's the most active toxin against Colorado potato beetle from Bacillus thuringiensis. And what we found in these bioassays is that there is a small difference, about 3 to 4-fold difference in the LC50, but the confidence intervals are almost overlapping. And for some of these doses, there was no significant difference. So we consider that there's really not relevant cross-resistance to Cry3AA in these double-stranded RNA resistant insects. Now that we had characterized the genetics and the cross-resistance in this strain, we were interested in looking at what is the mechanism involving resistance. Because these insects are cross-resistant to an alternative double-stranded RNA, that tells us right there the target mutations are probably not involved in resistance. And so we concentrated on other potential resistance mechanisms, such as an increase in nuclease degradation of the double-stranded RNA, reduced uptake of the double-stranded RNA, or potentially blockers or mutations that could affect the RNAi machinery in these insects. So when Swati looked at the nucleases, she looked at both regurgitate and gut fluid from larvae of the GC susceptible and the CS resistant colony, degrading the VATPase double-stranded RNA that we're using for selection. And what you can see here is that we did not find any significant difference in the speed or in the amount of degradation of these double-stranded RNA between larvae of these two different strains. What you see here, this little color, is just that we had to compose a figure from many different gels that were run at the same time. So all these results can be compared side by side, even though they have different colors in the figure. So nucleases were not involved in resistance. We then look at the RNAi mechanism and its induction by the double-stranded RNA, looking at the GC susceptible colony, the CS resistant colony, and the MET, which again is another susceptible colony. In both the GC and MET strains, we saw that 24 hours after ingestion of the double-stranded RNA, there was a reduction in the level of transcripts for the target RNA, which was the VATPase subunit A. In contrast, in the resistant insects, what we observed was an upregulation of this target gene, as you can see here, all the way to 72 hours post-ingestion. This upregulation for target gene has also been described in other reports that have been published, even though the mechanism is not known, but definitely this upregulation of the target gene can overcome the silencing induced by RNAi and contribute to resistance in these insects. We have also been able to look at the double-stranded RNA uptake in these insects. In this case, we use fluorescently labeled double-stranded RNA. We look at the midgut of susceptible and resistant larvae. And what we see after 1 hour is that when we look at the tissue and the susceptible insects, we can see accumulation of these red fluorescently labeled double-stranded RNA in the midgut tissue compared to the level of accumulation in the resistant insects, as you can see here in the bottom. In fact, it looks like most of the double-stranded RNA in the resistant insects is either accumulated in the apical surface of the gut facing the lumen, or even in endosomal vesicles, as you can see here. We have also been able to confirm these results using sequencing of small RNAs from these tissues. So Swati extracted small RNAs and sent them for MySeq sequencing, and then look for 21 base pair small interfering RNAs that were produced from the double-stranded RNA. So what you see is that in the susceptible insects, there is this increase in the number of these small interfering RNAs that are produced when the insects are treated with the double-stranded RNA, but that increase does not happen in the resistant insects. So in summary, the take-home message for all the research so far is that we have shown that CPV can evolve high levels more than 11,000-fold of resistance to double-stranded RNA delivered as a topical spray or a foliar covering. Cross-resistance is present to alternative double-stranded RNA targets, but not to Cry3A toxin, which mirrors the results that were observed with Diabrodica virgifera, with rootworm, and suggests that these two different pesticides can be used in combination. Resistance is transmitted as an autosomal recessive trait, and in CPV is polygenic, in contrast to the at least single locus. We don't know if it's monogenic or polygenic in Diabrodica virgifera. There is no altered nuclease activity that participates in resistance in CPV, but resistance is associated with both an increased target gene expression and reduced uptake of the double-stranded RNA into the gut cells. Our current work is focused on identifying the resistance genes involved in this resistance. And for that, Swati is now going through analysis of bulk segregant analysis and RNA-seq data. The goal is to try to identify resistance alleles that we can go back to natural populations and try to determine their frequency and presence in those different populations. And Swati is also testing for cross-resistance to small molecule pesticides that are used to control CPV, to see if there's any risk of this resistance affecting this control. With that, I would like to finish by acknowledging other collaborators that have participated in this research, Jody Beatty and Jim Baum from Monsanto and Bayer, which had relevant contributions to this research, as well as a number of collaborators that collected all the CPV beetles that we used in generating the GC colony. I would like to thank Monsanto, now Bayer, for initial funding that supported most of the aspects that I have shared with you today, as well as for providing enough double-stranded RNA for us to select this colony, which is not a simple thing. And also would like to acknowledge current funding for this project, which is provided by the USDA BRAC program. And the current funding is also looking at potential risks when comparing resistance to transgenic delivery versus topical delivery of the double-stranded RNA in coral potato beetle. So with that, I would like to thank you again for your attention and looking forward to the discussion session.
Thank you very much, Juan Luis. Maybe just to keep in time, I will just stick to one question. I think the first one for you is from Geert. Do these resistance strains show any other phenotypes, differences in fecundity, general health that has changed?
Yeah, that's a great question. Thank you. So I didn't include that information here. We actually had looked for fitness costs, and we do not see any fitness costs in our colony. In fact, Swati and Jim have told me at times that it grows more healthier than the regular susceptible colony, the GC colony. So we have not really observed any significant differences in fecundity or growth.
Okay, thanks. There are some questions from Guy, but if it's okay, we'll discuss them during the discussion afterwards. We're already a few minutes over time. Sure. So our last speaker is Rohit Sharma. He's from Ghent University, from the lab of Guy Smaha. Rohit will present his recent work on RNAi in the stingbug in the Zara viridula. The floor is yours, Rohit. Rohit, we can't hear you. I can't hear you in any case, but your microphone is off.
We can't hear him or see him.
We see the presentation, but we don't hear and see nothing.
I'm on mute, sorry.
Yeah.
Okay.
No problem.
Sorry, okay. So I will start from the beginning again.
Can you also turn on your camera, maybe?
Yeah, now the camera was on. I could see him when he was speaking.
Okay, so thank you. And thank you for giving this opportunity to present my research on this platform. And I'm a final year PhD student at Kent University, and I'm working on RNAi in Zaravidula, which is also known as subunit stick bug. So before going to main research, let's have a quick look on this pest. So this is a Hemipteran, belongs to a stingbug family, Lepidopteran. And as you can see in the map, it is distributed worldwide, but an important pest in the countries where soybean is majorly grown. And also it has 5 different life stages and feed on all the parts of the plant. So this pest not only just reduce the yield, but also affect the quality of the seeds. So when we start this project, so we have the information that can you see in the graph. The white bar represents the SGSP, Southern stick bug, and you can see that here the information was that only all the core RNA machinery are functional in this pest, but there was no information that whether RNAi is functional upon the delivery of Dsrna. But in other stink bug species, Schistoceros and Heliomorpha, they have already confirmed by oral delivered or microinjection delivered Dsrna that this RNA is functional, both by oral RNA efficacy and microinjection efficacy. So our first objective was to identify whether RNA is functional, and if it's functional, then what is the RNA efficacy. So here we use the microinjection approach. So we use this RNA of the essential genes first, and then we use the oral feeding bioassay to confirm the oral RNA efficacy, where this RNA of the essential genes were mixed with the artificial diet and the insect were fed in the artificial diet. So in the microinjection assay, we screened in total 10 genes, and 67 nanogram of each genes was injected in the second instar of this pest, and then observed for 14 days. And we saw that the effect was divided in 3 different ranges, and there is first one where was no effect, and the mortality was not different from the GFP and the water control. And there was moderate effect where the mortality was between 60 to 70%, and then final where the mortality was above 90%, which shows the higher effect by microinjection. And further, we also confirmed the functionality by qPCR, where it shows that this mRNA is suppressed upon the injection. So from this microinjection assay, we selected the 5 best genes based on their mortality. And further, they were used for the oral feeding assay. So in the oral feeding assay, as you can see in the figure, the Dsrna of the selected genes were mixed in the artificial diet, and the final concentration of the Dsrna was around 300 nanogram per microliter. And second instar nymph were fed on this diet for 5 days, which was followed by feeding on natural diet for 9 days, and then evaluated based on the mortality. And you see that only 2 genes showed around 45 to 50% mortality, and it was significantly different from the control. And here also we confirmed the RNAi efficacy in the expression of mRNA of the essential gene. Here is alpha COP. So from both the microinjection assay and oral feeding bioassay, we see that that RNAi mechanism is functional in this pest, but there is a difference in the efficacy when this RNA is delivered by microinjection or by oral feeding. And there, so to investigate further, we perform the ex vivo degradation assay, as the Dsrna's activity could be one of the important factor that can degrade the Dsrna. So here, this is how we collected the different body fluid, saliva, hemolymph, and milk juice. So in this figure, you can see that the rostrum, the mouth parts, is inserted in the pipetting tip. So the saliva was directly collected from the rostrum tip, and the midgut juice and the hemolymph was directly collected from the abdomen cavity after dissection of the insect. And for hemolymph, it was directly collected by the microloader. And for midcut, the midcut was isolated from here and then centrifuged at the lower speed, and the supernatant was used for the ex vivo degradation assay. Then the Dsrna was incubated in the body fluid and then run on the gel after interval of time. So here we can see that the Dsrna's activity is high in saliva compared to the midcut juice, and you can see that after 10 minutes that Dsrna start degrading. And in the midcut juice also there is a degradation, but relatively lower than the saliva. On the other hand, in hemolymph, most of the Dsrna stay intact, but we also saw a band with the higher size. And this is something we didn't know, and there's a possibility that potential protein might have bound with the Dsrna. So here we try to identify the protein. So here the gel, and we excise the band from the gel and collect it in a tube and then homogenize it to take out everything from the gel. And for this, we centrifuge it and take out the supernatant from here. And then the supernatant was used further, and then and the water was evaporated from the supernatant, and then it was run on the SGS page. And here you can see that it was a protein about 70 kilodalton. And after sequencing, we found that it is a lipofurans. And if we see in the previous studies that the lipofurans have been found that it binds with the Dsrna when Dsrna is incubated in the hemolymph in the cockroach or desert locust. And in the same study, it was also observed that when the genes specific to the lipofurans were silenced, then it did not show any effect on the RNA response. So we stop here, and we further focused on our main barrier, which is this Rnase activity. So this is the same gel. It shows that this Rnase activity in saliva, and you can see that in adult the saliva could faster, but still we saw some motility. So therefore, to confirm further, we were interested to know what is the fate of this RNA in the artificial diet. So to confirm this, we fed the insect on artificial diet mixed with this RNA, and then the leftover diet was ran on the gel at 2 different time points, our 48 hours post-feeding and 24-hour post-feeding. And you can see in this here that there was certainly some degradation, but there was also a huge quantity of the intact Dsrna, which means that during the whole course of continuous feeding of this pest on the artificial diet, the intact Dsrna was available. And also, as this pest have a feeding habit where it secretes the saliva and then leak it, so it also can be concluded that the saliva secreted by this pest during the feeding on artificial diet was not high enough that can degrade the Dsrna. So here the possible reason could be that the availability of the Dsrna during the whole course of feeding on artificial diet mixed with Dsrna was the reason that we saw a mortality in the second star when we compared to the degradation in the saliva collected from adult. So here we see that in microinjection assay and the oral feeding bioassay, there clearly is a difference in the efficacy. And the possible reason that we saw that the saliva with the juice show a degradation due to this Rnase activity. So our next objective was to check whether the Dsrnase involved in the lower the oral RNA efficacy. So here we use the RNA of RNA approach. So first we have to identify the Dsrnase. This is NV because these are regular. So here, since the transcriptome sequence is not available in the public domain, so we have to identify by ourselves. So after identification, we also interested to know the expression level of this gene across the different tissues and the different stages. And for identifying the role of this gene in lower the oral RNA efficacy, here this is the RNA of RNAi. First, the insect were injected with the Dsrna specific to the Dsrnase gene, and then fed on the artificial diet containing the essential gene. And we also investigated whether these RNAs still able to degrade this RNA after collecting the saliva from the insect where this RNA was silenced. So first here, how we identified this RNA sequence. So we use the sequence read archives database of Neisseria vidula, but to obtain the full length sequence, we use the mRNA sequence of Heliomorpha, which is the other species of steelbug. And then the whole fragment was amplified by PCR, and for the purpose of sequencing, it was cloned in the PJET vector. And for further confirmation, we did a phylogenetic analysis. And here we can see that this gene is in the clade of the dsRNAs from other steelbugs. So this is how we identified the sequences of this gene. And here is the expression level across the different tissues and different stages. So in the developmental stages, we can see that the early instar, particularly second and third instar, there is a higher expression level. But when it goes to the later developmental stages, then the expression level goes more stable. But if we see at the first instar, which is significantly lower than all other stages, there is one of the possible reasons that we can correlate is that the first instar does not feed, and it's the first this pests start feeding on the plant from second instar onwards. So when the when these insects start feeding on the plants, only then this gene start expressing. So this is here we can conclude from this expression in the developmental stages. On the other hand, in the expression in the different tissue stages, different tissues. So here we can see that the highest expression in the salivary glands. However, there is expression also in the different tissues. So this high expression in the salivary glands can be correlated to our previous degradation assay from saliva, as we saw that in saliva showed the highest degradation of Dsrna compared to the mid-cultures and humanoids. So here we can conclude that the salivary glands are the main source of Dsrna's protein synthesis. So further, we use the RNAi concept to identify the role of these RNAs in the lower the old RNA efficacy. So first here, the dsRNA specific dsRNA was injected in the second instar and then allowed feed on the natural diet. So in the meantime, so that mRNA expression of this RNA gene was lower. And then the same insect were allowed to feed on the artificial diet containing an essential gene. Here we use Alpha-COP, and here it was fed for 5 days. And then it was transferred to 9 days. And after 14 days, it was evaluated based on the mortality. And we also confirmed the effect of suppression of these RNAs on the expression level of the essential gene after feeding on the artificial diet with this RNA of the essential gene. Here we can see that survival curve. So first I explain this one. So this is the black curve, is the main treatment that what I explained in the previous slide, and it showed the mortality around 65%. And when we compared this treatment with the control, where the insect were fed on the diet that contains the essential gene Dsrna, but where the Dsrna was not silenced, and we can see that here there was a certainly significant mortality in the treatment. So here we can conclude that the suppression of this RNA gene certainly improved the old RNA efficacy. And if we look at the other control, where this RNA was injected and fed on the GFP, so when we see the mortality of this green curve, so there is no difference in the mortality to the other controls where the insect were injected with GFP and fed on either water or GFP. So, so there is no mortality in this particular control, which means that this RNase is not an essential gene. And here in this graph, which shows that the mRNA expression level in the insect where dsRNA was silenced is significantly lower than the insect where dsRNA was not silenced, which again shows that it also has effect on the transcript level of the essential gene. And we were also interested to know the fate of silencing of dsRNAs when we inject it on the first day. So at the time when we analyzed the expression level of essential genes, so at the time also the dsRNAs was silenced. So from this we can conclude that certainly RNAi improves the oral RNA efficacy in this pest. And we also checked the dsRNA degradation in the saliva that was collected from adult where dsRNAs was silenced. So here in the lower two gels, these are the degradation assay with the saliva and milk juice that was collected from the adult where dsRNA was silenced, and you can clearly see that the dsRNA is more stable in both milk juice and saliva and milk juice when we compare with the control. So this degradation assay support our previous result of RNAi, where this RNA of RNAi improved the oral RNA efficacy. So in conclusion of this study, we can conclude that this RNAi mechanism is functional upon the delivery of dsRNA by both means, orally delivery and as well as with microinjection delivered. And the difference between the microinjection delivery and the oral delivery is due to mainly the dsRNAs activity in the saliva and milk juice. And one of the main important reasons is the expression of these RNAs, and we have found that there is only one dsRNase which is mainly expressed in the delivery glands. And also RNAi has proved that here this suppression of this RNase gene improve the oral RNA efficacy and also prolong the stability in saliva and the juice. So this was all about my research here, and I would like to thank everyone for participating and your attention.
Thank you very much, Rohit, for your nice presentation. So I think we still have some time for discussion now, around 20 to 30 minutes, I guess. There are a few questions, maybe some technical questions related to Rohit's presentation. Rohit, did you try mixing the COP Dsrna and the Dsrna in a feeding experiment, so simultaneously at the same time?
I did not do that because the main fundamental objective of our research was just to first identify that whether suppression of these RNAs can improve the oral RNA efficacy. However, it could be a good idea if we think about in point of crop protection, then that the mixing can be investigated to check whether these after mixing the dsRNA specific for these RNAs as well as the specific for the essential gene, that could lead improve the efficacy. So it is I think the question of investigation.
So you think that mixing these two dsRNAs in theory could actually become an application as well?
Yes, if we investigate, then possibility, if we saw the positive results there, then of course it can be considered and it can have a potential, but without any evidences, it's hard to say that whether it's positive or not.
Another question is whether there was only one dsRNase that you've identified in this species related to dsRNA nucleolytic activity.
Actually, we before to exploring how many dsRNAs there, so we use multiple sequences from the different species. So like 4 genes from Lepidoptera and 4 from, which is I think from the Spodoptera, and 4 from the desert locust, and 1 or 2 from and one more set from the Coleoptera, which is this sweet potato weevil. So we blast this in the SRI database, all the sequence of these RNAs from other species, and every time we just hit only one dsRNase. So according to our experiment, we could find only one. But if we compare with other stink species, for example, in Heliomorpha, where it has been only one dsRNase is predicted, and also in the Ischistus heros also there's only one, and as well as in the green winged stink bugs, Luciferase Activity also there is one. Yeah.
Okay, thanks. So maybe we can go back to some of the questions that were asked for previous speakers as well, the ones that I've had to skip. I think the first one that I've saved here is one from Guy. So he was asking Steve, so the mosquito larvae, they are filter feeders. It's intriguing to see the activity. And do these compounds appear in the water as particles?
Hi. We were feeding them in a concentrated solution initially, so they had nothing there but to ingest the material that was exposed to them in those trials. But in the actual feeding trials where we were killing them, the paperclips were mixed into a pellet mixed with other edible components that they like to feed upon. We weren't counting on it being capturing enough double-stranded RNA just in the water column.
Do these pellets kind of dissolve then in the water, or do they really nibble from these pellets? No. Or
they were agar-based pellets that that were on the bottom of the trays, and so they would be having to go down there and graze on them.
Okay. I have another question here. A few more questions for Geert. One from Jim Baum again. Any evidence for movement via stoma in leaves?
Yeah, it was on mute. No. We didn't try very hard, but we didn't saw any evidence for uptake through the stoma. We used special sprayers and treating, let's say, the underside of the leaf and so on, but no.
Okay, another question from Marco. Will encapsulation of Dsrna help for increasing the stability? So I don't know if this is in the plant or on the plant. I don't know if Marco is still here, if he wants to clarify.
Well, you can, yeah, formulations to deliver more efficiently the double-stranded RNA to the insect might help, yes. I don't know how formulated double-stranded RNA will move into a plant, however. Will it follow the same route? Or will it get stuck? At least from the potato plants, it's clear that it's quite stable once it is inside the plant tissues. Huh? So you don't have to protect it directly against plant degradation by plant RNases, but maybe, yes, formulations that can increase uptake in insects can do the job. Yeah.
I also had another question. All the assays you did with the cut roots and the cut stems, then afterwards for example, you feed these materials to CPB, which is a very sensitive insect. Did you also try this with other insects? Whether it works?
No, we didn't. We didn't, and yeah. These were kind of side experiments. We should have also tried other insects. Yes, but no, I can't.
Yeah, I was just wondering because I'd assume you need much more Dsrna to be taken up in the plant's material for many other insects. I have a comment from Michael Wassenegger. Okay, it's not a question, it's a comment. So your results were very similar to our data from trunk injections and petiole absorptions. Then a question from Anastasia. Do you think that insect feeding on roots has a similar effect on Dsrna uptake as cutting the roots?
Good question. I don't know. But yeah, maybe if you just damaged, if you clip roots, for example, you see an increased uptake. So maybe insect damage can also increase uptake. I don't know. That's a good suggestion.
Yeah, because I think there's also evidence that if you damage leaves by abrasions, that you can also increase Dsrna uptake. So I guess feeding by insects should indeed or could indeed also have the same effect. Rohit had a question for you. Was there any formulation used in the stem soaking experiment?
No, there wasn't. That was just an aqueous solution with double-stranded RNA.
Okay. Then a question from Guy. Thank you, Geert, for sharing. Co-parameter is important for uptake in the plant and transport. What is the impact of plant cell wall, size, thickness, composition? I don't know if you have any idea on this.
I think if you mean uptake through the leaf, we were not able to get it inside the plant. Uptake through the circulatory system in this kind of hydroponic setups, well, we only tested, let's say, potato plants and tomato plants. I can't say if probably there will be difference in different plants, but I can't really tell if difference in cell wall or other parameters makes a difference.
Okay. Of course, if anybody has any further comments or wants to ask any follow-up questions, feel free to add them to the chat. I'll try to keep that in view as well. I think there were a few questions also for Professor Pali. The first one, what nanoparticles are you using, if you can reveal? So I think this refers to the last slide where you showed increased RNAi efficacy.
We are trying different kinds of nanoparticles. It's a moving target. Most of them are based on polymers or lipids. Right now, we don't have a magic formulation that kills 100% of the larvae. This is work in progress.
These are already existing polymers that are out there already, or these are completely novel?
These are, we are following an approach of coating. So we mix double-stranded RNA with a polymer A, then take that, make the complex and coat with another polymer. So it's an approach where we're trying to take a layer by layer. And as I said, we are not there yet where most of the larvae die when we feed these double-stranded RNAs prepared using these nanoformulations. It's, we are working on it.
Okay. I have one question from Paul, who is asking, what impact does gut pH have in Dsrna degradation versus Dsrnases?
We have not specifically measured the effect of pH on these Dsrnases, but based on our work in a variety of insects, I think 4 or 5 different orders, looks like Dsrnases may have a wider pH range. They can work in neutral pH, slightly acidic pH, and alkaline pH definitely. So I don't think they are very sensitive to pH.
Mm-hmm. Yeah, this is also something that we actually saw in several of our assays, that just putting Dsrna at a certain pH, even if it's pH 10 or 11, it does not change the Dsrna, it does not degrade the Dsrna itself. What we do see is with Lepidoptera that a higher pH just makes the nucleases more active. So the degradation by nucleases is more rapid.
Yeah, that sounds right.
Yeah. And of course, also the pH will have an impact on if you use nanoparticles on your complexation, especially if you use electrostatic interactions between Dsrna and a cationic compound. Your pH will also affect the stability of those complexes as well.
That's a very good point. We have been experiencing differences based on which insect we treat. So based on the size, charge, and shape, the nanoparticles that work well in Lepidopterans don't work as well in mosquitoes or stink bugs. and vice versa. So there seem to be some sensitivities based on the cell types and pH and delivery methods used in these. Most of our work is right now focused on Aedes aegypti, Nazara viridula, and Spodoptera frigifera. We are seeing significant differences among these three insects.
Right. Then another question from Juan Luis. What Dsrna doses are inducing mortality with the nanoparticles?
That's a difficult one, because the way our assays are conducted, it's very difficult to quantify how much Dsrna these insects are taking. But we feed them 10 micrograms per microliter, and these neonate larvae don't ingest even a fraction of that microliter. We really don't know how much they are getting in.
I have a question from Salvatore to Juan Luis. Your conclusions were that both increased gene expression and diminished uptake might occur in your colony. This would involve different IRM strategies. Which way you think this is going to be adopted in case of Higgs?
Yeah, so this increased transcript levels is something that we only see in specific conditions. In the test that I just showed you in the presentation, that was feeding on a high dose of double-stranded RNA. When we feed the insects on potato leaves with a lower dose of double-stranded RNA, we do not see that increase, that uptake. That increase in transcript levels. So we're not confident at the moment that that is one of the mechanisms involved in resistance. We think that it may participate when you have a high dose of double-stranded RNA, but we're not pretty sure how it's contributing to resistance. And in terms of IRM, I think the results so far in rootworm as well as CPB for the case of transgenic plants suggests, pyramiding with different modes of action, because using different targets is not going to work. And also using refuge, since resistance seems to be recessive, similar to BT. So it's a similar paradigm to the BT model.
Okay. I have another question for Geert. Just out of curiosity, do you think, is there any systemic movement of Dsrna from xylem to phloem?
As I said, I don't know. We think it is not. We tried some experiments. It's kind of EDTA exclusion assay, but let's say the results were not convincing enough to make any judgment. But the fact that it is actually not remobilized to other parts of the plant suggests that it's maybe not entering the phloem.
I have another question for you. Why does Dsrna spreading works better in apex and young leaves?
I don't know. Maybe you have more circulation to these newly formed tissues because they have to grow, they have to develop, they have to get more nutrients than an already existing leaf, I would say. Is that a suggestion? I don't know.
I have another question from Paul, a question for the whole group. Can chemical modification of the RNA improve stability, targeting, or cell uptake be a feasible solution in agriculture as it has been in human therapeutics? Or is there no way to do this economically? I don't know if anybody wants to give some input on this.
I can try, based on what we know. I think it depends on the cost for this formulation. If we are able to find a cheap material that can enhance the uptake and stability and efficacy of these dsRNAs, that would be a good solution. And this cheap material has to be fairly benign too. If it is has any adverse effects on non-targets and all, then we'll have a huge problem on our hands. It is challenging, but I think it is doable because there are enough natural polymers out there. One could utilize them to improve RNAi.
And I think I'll just comment on that point that I do think that we didn't think that it would be possible to do large-scale production of double-stranded RNA for field applications, but we're now seeing that where there's a need, there's a way to make it happen. And so we see companies now that can mass produce double-stranded RNAs very cheaply. So I think that chemical modification of them would also be relatively cheap and would be a nice, easy add-on to those technologies. I see it as quite feasible.
A comment that I would like to make is that, you know, with all these technologies that increase the stability of the double-stranded RNA, it also comes questions of potential non-target effects or other risks that may emerge when you make these molecules more stable in the environment, or they may work better in insects that are naturally not susceptible to it. Just a comment.
Okay. I think one more question I have here is whether the basic gut pH of Lepidoptera is critical for Dsrna inefficiency. Just from our own experience, we see that if you buffer, for example, gut use of Lepidoptera at more neutral pHs, you see the activity of the nucleases go down, but there still is degradation. So even at the neutral pH, there still is degradation, but it does, I mean, the nucleases are let's say, perfected to work at a certain pH. And in the case of Lepidoptera, this is an alkaline pH. But it's not critical in my experience, at least. I have another question for Professor Pali from Zarel. You mentioned it's hard to estimate the Dsrna concentration that the larvae eat based on the setup. Would you mind sharing what is your feeding assay setup? So is this an artificial diet setup, or is it a leaf-based assay?
We have been using two different assays for neonates. One is based on sucrose solution. We dissolve Dsrna or formulation of Dsrna in, I think it is 10% sucrose solution. I may be a little wrong on the concentration. I haven't been in the lab for a while. So that is traced with a food coloring dye. So we leave these larvae around that drop of material, and we only pick up larvae that ingested Dsrna solution with the color in their gut. The second assay we use is we use artificial dye pellets. Those are impregnated with Dsrna formulation. In both cases, as I said before, it is hard to quantify how much they are ingesting. We know the starting concentration, we know they have taken up something, but we don't know how much.
Okay, thanks. And then I think the final question we still have is from Bruno. It's a difficult question, apparently. How far do you see the release on the market of new RNAi-based products to control insect pests?
It's not far. It's not far. I mean, yeah, because in the US,
it depends whether you look at a broader range of insects or if you look at very sensitive insects, I guess.
Yeah. So when you say no far, it's not so far, what it means? In 1 year, 2 years, few years?
So maybe some of our Bayer colleagues, if there's anybody can tell us, but you know, the transgenic corn is approved in the US, was approved in 2017 already for woodworm. And I know there are companies that are already undergoing registration in the US for topical double-stranded RNA spray. So I don't think it's that far for what Olivier was saying, for insects that are susceptible, where the RNAi works very well. For these other pests like fall armyworm and etc., that's, you know, it's going to require improvements.
Can I have an additional question related to the difference between topical application and endogenous stable expression, so what is called Higgs. Do you think that is more far and more difficult to have such different other possibility to have resistant planet?
So I can share just briefly, our project right now is using transplastomic potato plants, which seems to be a better way of delivering the double-stranded RNA with less higher accumulation of double-stranded RNA, but also protected in the plastid from any degradation. So I think that may be a technology that may advance very quickly in the future for some crops where it can be done.
So the GMO approach, it will be more difficult.
Well, the GMO approach has been the first one to advance to being registered and approved. So I'm hoping myself that the topical sprays are just coming up very soon. But I cannot comment on that.
Okay, thank you.
Okay, I have two more questions for Juan Luis. Unless anybody else wants to comment on this first, no. So one is from Guy. So to Juan, an enormous work to collect all these insects in the field over the different US states. Did you genotype these strains? Also, how many individuals did you collect per location? Do you know if the Dsrna resistant individuals, do they come from one location or strain?
That's an excellent question, Guy. Thank you. So yes, it requires a tremendous amount of work, not only to collect these beetles, but to keep all of them, to rear them. So when we started receiving these collections, we ran some preliminary bioassays just to see if there was any difference in susceptibility to double-stranded RNA. And we did not detect any differences between the populations that we tested. We did not test all the populations. We did not genotype those initial collections. Rather than doing what you're proposing of determining where the resistance came from, we're hoping that once we identify the allele, we can go back to current field populations and determine the presence and the frequency of that allele in those populations. So rather than doing at the beginning, we're doing at the end of the project. But that's a very relevant question, and it's very possible, at least in CPV in the US, it is known that there are specific regions in the US where the CPV tend to be more resistant to pesticides compared, for example, to the West, where they seem to be more susceptible. So it is possible that there may be some populations that hold a higher frequency of resistance alleles.
Okay, thanks.
Oh, sorry. How many individuals collect per location? So, yeah. So there were 9 different states. We have a total of 12 locations from those 9 states. And we collected, that's a difficult estimate, but I think it was a minimum of 300 individuals from each location at different times. Yeah.
Okay, thanks. Question from Anastasia, also for you, Juan Luis. How confident are you that Dsrna uptake is limited in the gut versus limited systemic spreading? To me, the image you showed looked like Dsrna was being internalized into the cells lining the lumen of the gut, but not spreading inwards. If so, your resistant insect line might be a good system in which to explore mechanisms of systemic spreading.
That's a great question, and the answer is we don't know. We know in rootworm, where there was reduced uptake, it was also using the carcass, observing the carcass. So we have not done that test. We have not tested if it's only in the midgut or is a systemic phenomenon.
Okay, if there are no more questions, I think Bruno, it's almost 7 here. I think this would be a nice time to wrap it up.
Yeah, well, I want just to thank everybody. Thank first Olivier for managing this really interesting webinar. I'm not an entomologist, so for me sometimes it was difficult to follow, but it is really, really interesting, all the work that is now running on this topic. And well, I'm happy to know that in the next future there will be the possibility to have new RNA-based products, and I think that we also hope that in Europe this will happen, and we are doing this work in order to this type of activity or this type of webinars also for promoting this initiative and make more people aware of the situation, stakeholders, and so on. In fact, also the next meeting webinars we are going to organize are addressed to these topics. So in particular, the next one that will be next week is on communication, and the week after, in March 4, it will be for the stakeholders. We are working on the defining the program of the stakeholder meeting, because we want, for example, to involve many people from the European Commission. So it's very difficult, but we are trying to do this one. Yes, we are at the hand. If people still connected, turn on the camera. I can take a picture, a photo, a virtual group photo. And as already said, everything will go on the web page. I don't know if also Anastasia, Marco, and Chao He.
And also the previous webinars, for those who haven't seen them, they're still available online as well. Some on fungal applications or biosafety registration, things like that.
Yes, yes. And is Pali, yes, and Chao He. Well, Guy, okay. Chao He, are you able to turn on your camera? So we have the full screen. I don't know if I think that there is other people, but they are in the second page. And I don't know.
Maybe if they turn on the microphone, I think they get moved up in the list, but I'm not sure.
Yeah, well,
anyway,
because at least I take only the first page. Sorry, and I have one camera. Well, let me take one picture now, and I can also take the second page. Yes. Well, thank you. I think that we can stop here. And wait a minute, that now it's full, the first page. So I have the full things for the first page. Okay, thank you very much. Stay tuned on the program of iPlanta webinars, because we will have other 3 webinars in the final conference planned. So you will receive message, keep following it, and yeah, if you have questions, you can interact with us, with Guy, and so on. And we really hope to be in touch for future possible, maybe collaboration, exchange, another meeting, and so on. Have a nice day, a nice evening, and thanks again for your contribution to iPlanta webinar. Okay?
Thank you all. Bye.
Bye.
All right, thank you for organizing. Bye-bye.
Bye-bye. Bye.
Bye.